Okay, so we're going to get started with the second session of the meeting, and the next talk will be given by Titus Brown from Michigan State University. And maybe I'll just give you a little when you have 5 minutes.
Sure. All right, so
Actually, it'll go to yellow.
it'll go to yellow when I have minutes. Why do I only have 25 minutes? Okay, so 5 minutes for questions, I assume. All right, so this will be fast. So I'm at Michigan State University, and I have a confession, which is that I don't work in this space. So I have somewhat of a weird background. I did modeling and data analysis in undergrad. I got heavily involved in open source software development and software engineering while ignoring my graduate student work, where I got a PhD in developmental biology, working with genomics. And now I'm a computer science faculty crossed with the microbiology department, where I'm doing data-driven biology. So you can sort of say pretty much everybody's confused about what it is I do and what I work on, and how I do things, and what students I have, and what grants I apply for. So it's a very fun, sort of swirling interdisciplinary space that we're moving into with data-driven biology. And my current work is really focused on the big data problem from next-gen sequencing data, which I think is now, I don't think is going to be very interesting moving forward. I think that most of the problems will soon be solved with respect to next-gen sequencing data and volume. And velocity. And the interesting questions are now exactly the questions this workshop is addressing, which is variety. And so I've been thinking for a long time very hard about how to do data-driven modeling and model-driven data analysis, which I view as sort of separate and complementary activities. And I think Stephen sort of covered about 30 to 50% of what I want to say in this talk. So if you sense a certain amount of repetition, I had to give my slides in last night, and I didn't have time to update them after his talk, so I apologize. So I'm going to try and give an intermediate level view, because I don't work in environmental health sciences. I'm going to try and give an intermediate view of the challenges and opportunities of data integration, and talk about what types of analysis and discovery we want to enable, what the technical challenges are, what the common solutions that generally fail to these challenges are, and where we might look for success stories outside of biology. And I'm particularly happy that I looked outside of biology, because Stephen covered inside of biology. And then I have some conclusions. So I was trying to come up with sort of the specific set of questions that a lot of biologists want to ask. And this is at a much lower level than this kind of stuff Stephen talked about. But the very basic sort of thing is somebody else just published a data set that's relevant to a gene or a chemical that I work on. Do I see the same sorts of dynamics in that other data set? Or slightly more complicated, I have a known interaction. What other genes are affected that are potentially downstream of that interaction, or that impinge on that interaction? And then bigger questions like, what does chemical X do to an overall phenotype? What's the effect on gene expression, altered protein localization, patterns of histone modification? All the parts list stuff that Stephen said was not going to work very well. But everybody wants to know it anyway. And then there are the bigger questions, which of course we really want to know the answer to in a health setting, which are what's the effect of genetic background? What kind of combinatorics are there between chemicals, between different gene expression levels, gene expression changes? What are common effects of this class of chemical? That kind of thing. And so I really like the Rumsfeldian ontology of known knowns, known unknowns, and unknown unknowns. So I didn't want to put that up, but you can sort of classify these things. Known knowns are relatively well circumscribed. We know how to ask those questions. Known unknowns are things we know we're interested in and can sort of feel our way towards. And the real challenge, I think, of integrating heterogeneous data sets is enabling us to ask questions that we don't know we have yet. And more importantly than enabling ourselves, it's enabling all the other people. And this is where the leaderboard stuff and this competition, the gamification approach that Stephen talked about, where you make the data sets, small test data sets available, and then allow people to submit algorithms. I think that's having an immense impact in data science in the IT world. And I think it's great to see someone's doing that in biomedical research. So what sort of general behavior do we want to enable? I think reuse of data by groups that didn't or couldn't produce it is very important, right? What's the point of spending all this money to sequence everything on the planet when only the people that generate the sequence are capable of using it? something that I've been involved in, and I'll show you an example of next, publication of reusable or forkable data analysis pipelines and models. So the Synapse platform that Stephen talked about, there are a couple other things. I generally lump this under executable papers and data reuse. So there have been a couple really cool innovations in this area recently. One is in code, and I think you and Bernie, who will be talking, I don't know, next or soon after, we'll probably talk a little bit about this. But all of the data for the mod and code papers is available. All of the scripts to produce the figures that they published are available on a virtual machine. You can go out there, and with a minimum of effort, you can reproduce essentially their entire computational analysis from scratch using the data and the scripts they used, right? And that's really important, not for the replication or reproducibility issue. I mean, it's great that you can go investigate what they've done, because I can take similar kinds of data sets, replicate their analysis on it. I can compare my own new analyses to the data sets they've published and do cross-comparison, evaluation of models and evaluation of data analysis scripts and pipelines. This is the kind of thing that, as Stephen said, they've been doing in physics, they've been doing in I think, chemistry for years, for decades. And in biology, we haven't really gotten on board with it because of the data-driven gap. There's a paper in microbial ecology that came out in, I think it was Nature, or ISME, where they provided an Amazon virtual machine with the software and data to completely reproduce an analysis. And then there's a paper out of my lab on digital normalization, a streaming online method for data reduction that also, again, you can go and there's step-by-step procedures, about 8 hours, and for, I think, $4, you can reproduce our entire analysis from scratch on an Amazon virtual machine and see exactly where we fudged the data, right? Which is the important bit. It's the important bit. And so we've been making use not of the Synapse platform, but of this really fantastic thing called IPython Notebook, where you essentially like a Mathematica notebook, if any of you have seen that. You have the code that loads in the data and does the analysis and then plots the results, and it's all in one place. You can distribute the notebook, and anybody can run it if they have the right software, which you can provide on Amazon. And then it just makes it possible for other people to take your analyses and fork them. This is an open source term or a software engineering term for taking a body of source code, making a copy of it, and then modifying it to do your own work. And it's GitHub, which Stephen talked about, is essentially enables very easy forking. So I think enabling that kind of thing is something that we really want to be able to do. I've spent a lot of my time re-implementing published analyses so that I can do comparisons of my methods on their data to their methods. And it's a waste of everybody's time and money. Time more important, in fact. We also want to support integration of data and models. There's this concept called a mashup that comes from the internet world, the Web 2.0 world, where you sort of, different people make different data sets available, and then people take them and smush them together and produce something new and wonderful and that you never could have predicted, and I'll show you an example. And then something that I got very used to when I was working in Steve Koonen's lab. So this is a guy who was a computational physicist. He's gone on to the DOE. Now he's at the Center for Urban Science and Progress, I think it is, in New York City, doing data-driven city analysis. And I worked with him as an undergrad, and we'd have these group meetings where everybody would bring out their raw data in the form of graphs, and Steve and the others in the room would take the graphs and draw on them and scribble them and try and rearrange the axes and hold them up to the light to compare the pieces of paper, right? And figure out what the right next analysis was. And there's no reason at all that I shouldn't be able to have someone come into my office with their data set and sit there and tweak it and analyze it and integrate it in new ways. And we just, and come up with what the next big effort's going to be. And we just can't do that with the approaches and the software and the tools that we have right now. And that's really where I think advances in hypothesis-driven science come from. Better hypothesis generation and evaluation prior to doing the hard work. So an entertaining digression, I had two choices from a guy named Virgil Griffith. So this is books that make you dumb. So this is an example of mashup. Facebook made available by college the list of the top 10 favorite books for that college. Virgil combined that with a list of average SAT scores by college, right? Not the sort of thing that I bet either data provider had in mind, right? And so what you can see, it's a little bit odd. You know, the high SAT score colleges like Lolita and 100 Years of Solitude. I was a little nervous about putting this up because the Holy Bible and the Bible don't rank terribly well. You know, I'm a little surprised that the Grapes of Wrath is, anyway, but you get the idea. there's actually probably a better known example of this called Wikiscanner, where you can go and cross-reference who has been making edits to Wikipedia pages with their source, their physical location. So you can see who has been making changes to which congressman's page based on from which office in the Senate, right? And this has led to some high-profile, cover-up, investigation of cover-ups. And so these are examples of mashups. You make the data available, you cross-reference it by smushing it together in whatever ad hoc way you can, and then interesting things happen, sometimes positive, sometimes negative. It's an interesting thing to look up. So what are the obstacles to this? And this is sort of the 1,000-foot view or below. There's syntactic incompatibility. So the first 90% of bioinformatics is always trying to figure out how to take your two different data sets and cross-reference them. Right? The IDs are always in the wrong format. Sometimes it's just a comma. Much more often it's I have the one true identifier here, you have the one true identifier here, and we need a mapping between them. And anyway, semantic incompatibility is actually a real problem. What does gene mean in your database, right? So there's gene, which is protein coding. There's gene, which is coding, which is transcribed sequence. There's gene, which is locus of heritability. These things in different databases, they all mean different things. The genomics world has done a good job of collapsing this down. The rest of biology is still trying to figure out how to cross-reference with that. And then there's impedance mismatch, which is a technical term for basically saying you want to query your data in different ways. Sometimes you want to know all of the phenotypes that are associated with a given gene. Sometimes you want to know all of the phenotypes associated with this interval on the genome, with variation in this interval in the genome. Those are very different types of queries. Different kinds of databases support them in different ways. And unfortunately, or for better or for worse, SQL databases, which support a very limited set of sort of Excel-like spreadsheet queries, really dominate because basically for historical reasons, despite potentially better alternatives, graphs or object databases. And so these databases offer different kinds of queries, different kinds of data exploration, and they inherently limit how it is you explore and cross-correlate your data sets. And so just thinking more broadly about how to bring a lot of the CS research that has been done on these things into practical impact on biological data sets, I think could be quite valuable. There's also data volume and velocity. And the real point here is, data volume and velocity are only problems because they make everything else 10 to 100 times more difficult, right? So we already have these problems that have been known for probably since World War II, or well, since for millennia, frankly, in libraries, but all of these different cataloging systems and cross-referencing systems. And data volume and velocity just makes it that much harder to resolve these problems because now you can't do the queries quickly. So, I should say that when I asked around among friends for what examples of where these problems came in, and health records was one of the top two examples of catastrophe land, where it was just very hard to cross-reference. And then unstructured data, which gets back to both publications and health records, most of our information is locked up in PDFs, right? PDFs are like the worst possible format for computers to explore because, for all sorts of reasons, including basically none of the information in PDFs is readily available. It's just like a black box, like the environmental health records. You have to do all sorts of tricky stuff, and Google's really good at this, but it's an additional barrier. There's all of the syntactic and semantic incompatibility combined with sort of just general formatting and document availability problems. And so these are the obstacles, and the solutions basically boil down to entity resolution, which is you have a common identifier. And basically there you need a global naming system, and everybody has their own, which is kind of a problem. Top-down imposition of structure. So this is where one person says, this is the database. We've spent a lot of money on it. You will use it, or you will not get funding. Very popular approach. And it limits flexibility, and it reads well on grant review panels, right? Like, we've already sunk $10 million into this database. You're damn well going to use it. Okay. And the problem with this is that it sort of limits future flexibility because you have to go through that centralized. I'm not a big fan of centralization. I haven't been a professor long enough. So, it prevents use of unstructured data. It's kind of heavyweight. It requires consistent high levels of ongoing funding to maintain that central database. And there are a few places now that do it really well. The genome centers, the genome databases, Ensemble and David Hauser's group and NCBI all do fantastic jobs of this. But they aren't necessarily the most agile of organizations by design. right? They're supposed to be stable and slow to some extent. Although I don't think Hausler believes that, but anyway. So, and then there's ontologies to enable correct communication. So gene ontology and phenotype ontology are sort of obvious examples of this. This is centrally coordinated vocabulary. And not only is this slow to develop and hard to get right and often very expensive, it doesn't really solve the unstructured data problem. Nothing does, just in case you're wondering. But you end up in these sort of weird philosophical discussions. You know, well, now that we know that splicing is coupled to transcription, do we say that splicing factors belong in the ontology under transcription factors or not, right? And you end up having these like half-hour-long conversations in meetings where 2 or 3 people are very engaged in splitting hairs. And that may, in fact, be very important at the theoretical side, but how to balance that with practical applicability of ontologies is I think, very hard. I avoid ontologies like the plague because I can't stand those conversations. And then there's what everybody actually does, which is winging it. Ad hoc entity resolution. And this is a very common solution, right? You go out, you write a bunch of scripts, you remove all the annoying commas, you throw away all the data that doesn't agree with each other, and then you publish that, an analysis based on that. And that doesn't really work that well. And I think the real point is it's not capacity building, and it's not a foundation for further work, right? My ad hoc script is not something you can use tomorrow. And what we really want to move towards is a situation where as many of these problems as can be solved reproducibly get solved reproducibly. So we just can stop worrying about this this is all boring stuff. I spend my life doing it, and I'd rather not, right? We want to solve biological problems. That's kind of the point. So XKCD is a wonderful comic. Are better standards a solution? So everybody likes to propose a standard, and there are 14 competing standards. That's ridiculous. We need a new standard that encompasses all of those use cases, and soon there are 15 competing standards. And this actually happens quite a bit. And the only way this gets shut down is by fiat, when funding agencies and review panels refuse to fund anybody that's not using one of the 14 established standards, or one of the 3 reviewers' favorite standard, depending on how it goes. Okay, so my technical goals, and this was a very tough talk to arrange because it's not my usual research talk, and so I wanted to rephrase the technical goals for my talk are, how can we best provide a platform or platforms to support flexible data integration and data investigation across a wide range of data sets and data types in biology? And I want to avoid centralization. I want to support federated rollout, that is, of new data and functionality. I get a data set, I have a nifty new idea for analysis or modeling or integration. I just want to do it. I don't have to talk to the master guy about it. Provide flexible extensibility of ontologies and hierarchies, sort of a gray zone between this is the established ontology and this is stuff we just made up because it seems to work today, and a way to move between those. And then support, and I think this came up in Stephen's talk too, support a diverse ecology of databases, interfaces, and analysis software, right? How is it that we can, I ran into this at an NSF workshop where Lou Gross was there also, where somebody said, we just want to have one piece of software that everybody uses to do this thing. BLAST is the canonical example. Why would you ever need anything other than BLAST, apart from the fact that it's slow and doesn't work that well? And what you really want, what you really want is an ecology of competition, where you acknowledge that different tools have different trade-offs in time and speed and sensitivity and specificity. You let them compete. It gets more confusing for everybody, but at the end of the day, you end up with a much more robust system. And that's really where I want to go. So thankfully, I didn't know what Stephen was going to say. Thankfully, I looked outside of biology because he's pretty much covered inside of biology. And I really wanted to look for domains with large amounts of heterogeneous data that are continuously increasing in size at a very fast rate, are being mined effectively on an ongoing basis, have widely used programmatic interfaces as well as web interfaces that support these mashups and other cross-database stuff, and aren't accidental. That is, people set out and said, we're going to do this. How do we do this, right? So anybody in the audience have an idea of what I'm going to say next? You haven't seen my slides. So what company is taking over the world doing exactly this? It's not Google. Amazon. So Amazon has over 50 million users, probably over 100 by now, over a million product partners, billions of reviews and other pieces of information about what it is you like, your click trail. I looked at this book, I followed it to this book, I followed it to this. They track all of that, right? They have dozens of compute services. I don't think people entirely appreciate just how big Amazon Web Services and their cloud computing really is, and the vast variety of compute that they provide. When Amazon Web Services goes down, I think 10% of internet companies go down in some way, shape, or form. And they've explicitly adopted what they call a service-oriented architecture that enables internal and external use of this data. And one of the tricks is that they don't differentiate between internal and external use of the data, which I'll get into in a sec. So when you go to the Amazon.com webpage, over 150 different independent services, databases, and queries are executed to pull together that page. What it is, who you are, what it is you last looked at, what they recommend today for you, what their specials are, et cetera, et cetera, et cetera. That's not typically the way people build web pages, right? That's on the fly, fast, profit-driven integration of lots of different data sets, of vastly different data types. And the frustrating thing about Amazon is this is already incredibly hard to do, and they sort of routinely and casually deploy new services and functionality on a regular basis. They're out innovating everybody else in the cloud computing field. And they're doing it stably and robustly, having essentially solved, or at least the hardest computer science problem in existence, one of the hardest computer science problems in existence, which is the problem that we all happen to want to solve. So how have they done it? The sources for this are two documents. The Platform Rant by Steve Yege, who's someone who worked at Amazon and then went to Google and told Google that they were doing it all wrong, and then posted it, which was an interesting strategy. And a summary of a bunch of interviews and so on with Amazon people at highscalability.com, which focuses on questions of scalability. Amazon's doing this for scalability reasons and profit reasons, not for scientific, problem-solving reasons. But and they're both long and techy, but the first is really entertaining, and I highly recommend them as things to skim and sort of see. This is how tech geeks speak when amongst themselves. And basically, there's only 3 core principles, and these are mandates that come from the CEO. You get fired if you don't do this. All teams must expose data and functionality solely through a service interface. All the communication between the teams happens through that service interface. If you want to provide data to somebody else, it has to go through an interface you provide them. There's no back doors into the database, no surreptitious emailing of text files. It's all formal. And basically, all of these service interfaces are externalizable. They're things that the outside world can use. Now, this is really important when you think about things like HIPAA, which I'm not supposed to mention, right? You think, Amazon has your credit card information, right? People care about that probably as much or more than they care about their medical records, at least at a sort of daily basis. And Amazon has figured out how to do this, right? They have a whole security infrastructure. They have a whole sign-on system. It all works. They confront problems that are just as hard as the problems that we're confronting here, except that they have to make money off of it on a daily basis as well. And the colloquial term for this is you should eat your own dog food. So the best recommendation for your own dog food is that your dogs eat it, probably not you eating it. And so what this really means is you use what you produce. If you have the courage of your convictions, you use what it is you produce. And we're really bad at this in academia. We're really bad at making these things available in the way that we make use of them for publications. That's why the executable data stuff, the executable paper stuff is so important, I think. So basically, when you design and implement the database and database functionality to meet your own needs, your needs surely have significant overlap with other scientists in the field. If you want to adapt to a research setting like we have here, you probably won't be able to have hardcore molecular biologists who are also good database programmers, so they should work closely together. Everybody buys books, it turns out. So Amazon has that advantage. When they're designing things, everybody sort of knows what the customer experience should look like. Here we have really a diverse set of expertise and backgrounds. And I think you want to make sure that what you're doing is useful. Genome databases have done a really good job of this, but in a centralized model. And I'll just put up this little slide. So this is a gap between what happens in software development if the customers aren't integrated in the development loop. There's how the customer explained what they wanted, which is a swing, what the customer really needed, and all of the stuff in between what the customer explained and what the customer really needed. And I particularly like how the project was documented. Not being a big fan of theoretical analysts, how the analysts designed it. So, all right, so what does this look like in practice? And this is really something I've been thinking about from the context of what I'm doing, which is sort of investigations into how to deal with expression-level data and cross-compare between microarrays and different mRNA-seq experiments. And And the idea really is you have a bunch of different boxes that have a bunch of different data and services on that data, and they interact in a very loosely coupled way. So you have potentially completely distinct services that offer expression normalization and can pull from a variety of your primary data sources. Maybe a particular normalization technique cannot deal with microarray data, in which case it can't pull from that source. But the normalization is where the integration happens, and it can then be pulled back into a metabolic model or used for differential expression query or just for plain data exploration. You can keep a separate database of, for example, chemical relationships or gene ID translators. And all these exist as separate services. They can be centralized in the sense that they may all be running on the same compute equipment underneath, but the idea is to think of them as logically and physically distinct so that you don't accidentally write a normalization routine, a data normalization routine that relies on internal details that aren't exposed generally. And the underlying acknowledgement of this view is, the underlying thing that this view acknowledges is that everything's changing all the time. So Carolyn gave me a very specific mandate, which was not to talk about data types changing, and I can't. Because Illumina changes their sequencing technology every other day. Microarray people, when they're not going out of business, change their technology every other day. Their formats change. The biases and idiosyncrasies of the data change. Normalization is a moving target in the sense that we still do a really bad job of expression normalization. And there's active research going on in this. We do a bad job of isoform counting. All of these things are going to be systematically improving over the next couple of years. And we don't want to have to completely rewrite this in the face of a change to this. So everything's changing all the time. The term that the Amazon grant used was embrace chaos. Not something program managers often want to hear, but in practice, when you look at what's happening with the velocity of data and the scaling of the data that we're getting, it's clear chaos is inevitable. So we should figure out how to prepare for it in sort of a resilient sense. I'll make a few quick points. So that's really the main point, I think, is that go towards a loosely coupled thing that allows you to roll out new services. I could publish a new normalization routine, copy this box, make a new one that has the same interface but implements a different algorithm, and allow people to do straight up cross comparisons by doing the same queries on two different boxes and comparing the results, right? That would be tremendously valuable from the perspective of publication, I think. Okay. We know how to do this. The open source and agile software development communities have developed approaches that aren't typically used in academic software development and should be. Developing services in small groups that include customer-facing developers helps make sure that you're developing the right thing. And then implementing all of the cloud stuff, it gives you flexibility in tools, approaches, and implementation, and is really helpful. In my remaining few minutes, I just want to talk about data, about modeling as well. So something else that we have done a really limited job of in biology is to combine modeling with the data that we're generating. So there's two different ways to look at it. You can do data-driven modeling where you determine connections and parameters and influence based on the raw data, which is what Stephen was really, I think, arguing for. And then there's the flip side where you do model-driven data investigation. You take your known model and you look for outliers in the data that doesn't fit that model. Those are likely to be interesting. They're either going to be interesting biology or interesting for your model. One or the other, or interesting for data analysis, right? One of the three is wrong. One of the three is novel. And this is how, everybody likes to talk about the CERN accelerator data. This is how the physicists work. They build a model, interpret the data using the model, throw at everything that they've seen before that's uninteresting, and then just look at the new exciting stuff. And there's a lot of problems with adapting this approach to biology, as I think Stephen covered very adequately. I just sort of wanted to cover, so in my graduate work, I worked on this network for early development in the sea urchin. We had about 150 genes by the end with the interactions between the genes. And the really cool thing was, when you zoom in on various aspects of the model, here's where nuclearization of beta-catenin influences endomesoderm specification. You can go in and understand, based on the model connections, you can predict exactly what a particular signaling molecule blocker is going to do based on the connections that are present in the model. And you can compare that prediction with what actually happens and learn whether or not your model is sufficient to handle that perturbation. What was really surprising for me, so there's this model and it's complicated and we published on it, and it's not really a computational model, it's really more of a logic model that we use and we pore over and draw things on. And the thing that was really surprising for me about it is how quickly the community converged on this model as a common language. So there was no longer a question of what molecule are we talking about and what interaction are we talking about. We're talking about this one. I just happen to think you're wrong. Right? And as a common language for disagreement, as a common language for where we agree and where we disagree, to highlight the things that we should be focusing on, it was actually an incredibly amazing transition within the sea urchin field. Within a year or two of us coming out with this model, the entire community was arguing about the model rather than about the names of molecules, which was a welcome change, I guess I would say. Okay. Very quickly at the end, there are a lot of social obstacles, and I think these are the real problems. And again, as I said, there's a lot of overlap with what Stephen said. We don't train biologically aware software developers, and we don't train molecular biologists in particular. Most biologists, I'm very familiar with molecular biology training programs in computation. So I often get the comment, look, I just want the answer so that I can go do the real work of figuring out if it's real biology. And it's like, well, it's not, it's not quite that simple. It's an interesting challenge. And then, of course, incentivization. You don't get publications or grants or much respect for sharing data and source code. We don't really understand the patterns for useful data sharing, which I think is a lot of what my talk is about, is we don't want to constrain in advance what we expect people to do with the data. We want to make the data as openly available as we can within the constraints of the regulatory burden. A few other places to look. I'll just leave these in the talk. NEON and other NSF centers are confronting this. SBML is a modeling language that enables interoperability of models. And then software carpentry is something I'm involved with that runs free workshops on effective use for computation for science. So look it up. We'll come run a workshop. You just have to pay for somebody the flight. And then we'll come out and run the workshop for you. Okay, so my conclusions is I think we really need what Amazon calls a platform mentality. We're providing the data and functionality on the data as part of a platform for data investigation. It is not our job to constrain the data in ways that we think you will find useful. It's our job to make it as useful as possible and then let it be after that. Agile and end-user focused software development methodologies have worked really well. And I think one of the big failings of research science in general, computer science in specific, and bioinformatics as well, as part of that, is that a lot of these details have been explored, if not solved, in the internet world. And we completely ignore it. In fact, there's a steady transition of people from the computer science world into the high-tech world, because that's where all the interesting problems are being solved. We don't have the scale or the nomenclature and background in computer science to work with that. And so it's, we shouldn't be solving problems that have already been explored. And then data is most useful in the context of an explicit model. I think models can be generated from data, and the models can feed back into the data gathering. There are things I didn't mention. The only one I really wanted to point out is that all of these technical problems are much easier to solve than the social problems. The social problems are the problem. We don't have the developers. We don't have the users. We don't have the mindset. So we need to train those people. So thank you.